So, hi. Our question is how can proteins of gut bacteria affect the growth of colon cancer? So, I'm Angela Xiang from Portola High School.
I'm Francesco Donato from Leone XIII High School.
And I'm Harsim Rabal from Moore Park High School.
So, how do certain proteins of gut bacteria affect the growth of colon cancer? Gut bacteria is one of the many types of microorganisms found in gut microbiome of humans. Scientists have found that gut bacteria can promote several host cellular multiplication and survival pathways, contributing to the appearance of colorectal cancer. One issue regarding colorectal cancer is the location of the proteins. It is not clear if the location of the proteins in cancer-causing bacteria affects tumor growth. We propose further studies and experiments on gut bacteria that are associated with tumor growth in the colon in order to determine how they cause cancer. We plan to place healthy colon cells in vitro with sample bacteria and use several microscopy techniques to study how the different bacteria species cause cancerous growth. We conclude that each cancer-causing bacteria will promote colon cancer with different methods.
There are several bacterial populations which directly influence tumor development in the colon. The main three are Escherichia coli, enterotoxigenic Bacteroides fragilis, and Fusobacterium nucleatum. This is relevant because if we are able to compare the movement of the proteins from the bacteria to the colon cells, we will be able to determine how these proteins can cause the proliferation of the colon cells. Answering our research question, we will contribute associated with three main advantages. The first one is that we will have more information about the colon cancer, and thanks to this, the second one is that new treatments can be developed. The third one is that if we can develop those treatments, we can help to cure more people all over the world.
So as of right now, researchers have utilized several microscopy techniques to study and confirm which bacteria in the gut microbiome have the ability to influence cancer formation. Certain bacteria can stimulate cancer formation by blocking immune effectors that normally inhibit tumor growth. For example, F. nucleatum inhibits killer T cells from functioning properly in the immune system and attacking pathogens, therefore helping cancergenesis. The ETBF, also known as enterotoxigenic Bacteroides fragilis, is a bacteria that produces a toxin known as BFT, which directly causes colorectal cancer. Another cancer-causing bacteria, E. coli, produces the toxin colibactin, which mutates DNA in colon cells, and depending on which genes are mutated, the colon cells can grow uncontrollably, resulting in cancer formation. So for our experiment, we plan to observe test tube samples of each of the three cancer-causing bacteria in vitro with healthy colon cells and compare the cancer growth in those tubes with 3 test tubes also in vitro that only contain specific proteins from each of those bacteria. We chose this experiment as it will allow us to determine if the location of the proteins in regards to the bacteria will affect colorectal cancer growth. Our hypothesis is that the protein of each bacteria are the sole cause of cancer's growth, so regardless of if the proteins remain inside the bacteria or are secreted, cancer will still appear. The first part of our experiment is to identify which proteins in each bacteria cause tumor formation. In order to confirm that our samples are of the correct bacteria, we will utilize gene sequencing and compare the entire nucleotide sequence of previously identified samples of each of the cancer-causing bacteria with our new samples. Then, to find which specific proteins in each of the 3 bacteria cause tumor formation, the polymerase chain reaction can be used to cut off all DNA that does not code for cancer-causing proteins and can then amplify the specific protein sequence for comparison with known sequences of cancer-causing proteins. For the actual observation, we propose to grow the colon cells in vitro in 3 test tubes and then add the 3 types of bacteria, E. coli, F. nucleatum, and ETBF, to study if and how proteins move from the bacteria to the colon cells. In order to be able to see the proteins, the gene that expresses green fluorescent protein will be added to the proteins of each bacteria. Using a fluorescent microscope, the fluorescent protein's localization patterns can be observed in order to determine whether or not the proteins of each of the 3 bacteria were secreted into the environment. The fluorescence of the proteins makes it easier to track if proteins were secreted from the bacteria and were able to bind to colon cells. But for confirmation, the cancer-causing fluorescent proteins can also be removed from the bacteria and isolated in vitro to determine if cancer growth will still occur. The proliferation of cancer cells in each of the test tubes containing bacteria can be graphed every 12 hours in order to have comparable data with the test tubes that only contain proteins. If the proteins are able to cause cancerous growth in colon cells without the presence of bacteria, it is clear that the proteins are the sole cause of colorectal cancer. If the proteins do not have any effect on the healthy colon cells, then it is reasonable to assume that the specific bacteria cause proliferation and the cancer growth. Finally, to create a solution, it would be reasonable to compare the path each bacteria's proteins take in order to determine if they are able to directly travel to the nucleus of colon cells or are transported into the body cells by vesicles.
There are many challenges that are opposed to our project. The first one is that proteins cannot be tested in vivo. In fact, with our instrument, they can only be tested in vitro. The second one is that it's difficult to study proteins with the microscopy techniques that we have already. The third one is that even if price is making even if price to make this experiment is reducing, it is extremely costly. The fourth one is that find what proteins can cause colon cancer is very time intensive because there are many proteins in every bacteria in our body, and for every protein to study, we have to study for 30 minutes every one.
All right, so overall, our project aims at finding whether proteins of bacteria cause cancer or if the bacteria itself causes cancer. So in the future, we could take some steps such as combining different types of microscopy techniques such as electron microscopy and atomic force microscopy in order to better observe the protein and the gut bacteria. We could also develop an antibody in order to bind and destroy the cancer protein. And we could also create sort of a pill or a capsule that can destroy vesicles carrying cancer proteins by traveling through the digestive tract and binding to cancer-causing proteins, which would inhibit cancer growth. So this experiment is important because it would take us one step closer to finding a cure for colon cancer. It would help us to get a better understanding of the relationship between gut bacteria and colon cancer. It would lead to further discoveries of more protein found in bacteria, and it would also it could also help us to develop new methods and to prevent or treat colon cancer. So these are our references.
Okay, so I guess I'll start with the first question. So for your future directions, so you talk about using cryo-EM and AFM. Can you be more specific as to what you would use each of those techniques for in terms of what exactly you're looking at?
So cryo-EM and AFM would help us to get a better look of these proteins. These proteins inside these bacteria are extremely small, so it would help us to look at these live samples, and we could also stick them under there to see exactly what the growth is, and we can look at it and observe it over a long period of time.
So would you be trying to track like the location of the protein, how many proteins, what the protein is doing? Like what exactly is it about the protein that you're looking at with these techniques, I guess is what I'm asking.
We're trying to find the location of the protein, so the different types of microscopes would help us to better take a look at these very small proteins.
And also to add on, atomic force microscopy can be used to study like the replication of bacteria. So if some minor adjustments were made, it could be used to study even smaller proteins and see how they proliferate and how they grow when grown in vitro.
So you explained very well how you will use positive controls and negative controls in your experimental design, and how you will use fluorescence microscopy to follow either the protein or the bacteria. You also mentioned that you will use colon cells, right, in your assay. And I guess my question is, how would you measure that these cells become cancer cells? What is it that is going to tell you that what you are doing or adding to the culture is going to turn those cells into cancer cells?
Well,
and how would you measure it?
We can measure that by graphing how much the cancer cell or the normal colon cells, if they start to proliferate tremendously and they don't stop, then it's clear that they're becoming tumorous, and that indicates that there's cancer growth. So if we are able to record that data, it will show us how much growth is occurring and if it's linear growth or if it stops at some point.
You guys have done a great job. It's very impressive.
Thank you.
You mentioned that you want to use different kind of microscopy techniques to look at things. So just wonder, why do you want to look at them? How would that help you in this particular study?
Could you repeat the question real quick?
So you want to look at things, right?
Yes.
You guys, you're going to see things. So why is that important?
Oh, the importance of looking under the microscope would be with the fluorescent microscope, which we have tagged the proteins to become fluorescent. So we can track if the proteins in bacteria are excreted and bind to colon cells, or if they remain in the bacteria. And the bacteria will cause proliferated growth.
Yes, one more question.
So I also think this is a very great presentation, and as you guys mentioned, there's a time and a cost factor. One of the things I think you did really well was you went and researched where the current state of the art is, not just found bacteria that were associated with it, but putative mechanisms, right? And you selected these 3 proteins that you wanted to look at in particular, right? Then you also said you want to look at many, many other proteins, right? You want to scan if it's the proteins, all the proteins in the bacteria, for example. Except for the microscopy facilities we went to this week, do you guys think have you guys seen another facility that would be able to help with that kind of time-intensive process here at the CNSI?
Yes. You have asked a very intelligent question, and
I can short circuit it. It's just simply put, you guys went to the 6th floor and saw the screening facility.
Yes.
That's the type of facility that's dedicated to doing exactly the experiment you're talking about, where you've isolated all 100 different types of proteins. They can even grow proteins, each different protein in a well, suck them up, and test 100 proteins all at the same time in one batch, and that takes a half hour. And they can even do that proliferation assay you mentioned.
Yes.
And so this idea of coupling what you can and images. So the point is, they were taking the technique. If you as the researchers go to this light microscopy lab downstairs and develop an assay, the technology is already there to support you to try to answer those, can we do it in time and can we afford the cost? And having all of this in one building is one of these ways we can leverage all these different aspects of technology to develop what you guys learn into something actually productive that we can then translate into these results you're looking for. So I think it's this very forward-looking and very realistic work that can be done in a place like this. So.
Yeah.